Hello students, today we are going to revise this chapter genetics, some basic fundamentals. Just like the previous chapter, that is structure of chromosome, this is also a very new and fresh topic for you and which you find it very difficult, but again I am sure after 30 to 40 minutes you will feel that this chapter is very simple for you. Now the chapter is divided into three parts. First, the basics of genetics. Before we study about genetics, there are certain terminologies which is used in genetics, just like in nursery or junior class, before making a word or teaching you word or statement, they taught you alphabets. Same way, before we study the application of genetics or genetics branch of biology, we need to be thorough with the basics of genetics, that is terminologies of genetics. So first part is terminologies. Second part is the scientist, his experiment and his application in plants, how he has conducted the experiment, that is genetical experiment, and last its application in human being. So these are the three parts of the chapter. Fine. Now first of all, who introduced this branch? The name of the scientist is Gregor Johann Mendel. Full name Gregor Johann Mendel. Now Gregor is the title given to Johann Mendel, just like Mahatma Gandhi. Fine. So Gandhi's name is MK Gandhi, but he is called Mahatma because a tag is given. Same way over here, Gregor means most important person of the society, and this was the tag given to Johann Mendel. Johann Mendel is a father of genetics. Who is called as father of any branch? The person who come up with the basics of that branch. It means Gregor Johann Mendel has laid down the foundation for conducting genetical experiment. So he is known as father of genetics. Next, there are a few things about Gregor Johann Mendel which you need to remember. First of all, when he was born on 22nd July 1822. Fine, he was born in 18th century. So it's a story of 18th century. Second, he was born in peasants family. Next, where he was born in a village known as Moravia, which was in Brunn, which is now known as Brno, and this falls in Austria. Fine, but now this place is in Czechoslovakia because after Second World War, Austria was divided into two parts, Austria and Czechoslovakia. So now it is known as Brno and it falls in Czechoslovakia. Fine, remember this thing. Next, where he completed his education. He completed his education in monastery of Brunn. Why he need to go to monastery? Because he was born in peasants family, very poor family. And his parents cannot afford his education. That's why by himself he walked into the monastery of Brunn and got his basic education completed. Fine. The members of the monastery, they thought that he is a much more intelligent person. That's why they sent him to University of Vienna for his higher education. Fine. University of Vienna for his higher education. Now the student, he was a person who was very wise to the society, wise to the people who has done something for him. So instead of staying in Vienna and teaching over there or earning over there, he came back to the monastery and continued as a teacher and priest. Fine. He tried many times to become a teacher in Vienna, student, but over there, he could not clear many entrance exams related to the teaching. That's why he came back to monastery. Not that's why he was offered to teach in university without entrance exam also, but he rejected those offers and came back to the monastery of Brunn and continued as a teacher and priest. But in the year 1884, he died. Fine. He died as a abbot. Fine. You have to remember, he died as a famous teacher and abbot. Abbot is a person hired to the priest. So this is all about Gregor Johann Mendel, who laid down the foundation of genetics, known as father of genetics. Next we are going to study is the basic terminologies which we are going to use in genetics. The next thing which we are going to study is terminologies. To be specific, it will be genetical terminologies. Genetical terminologies we are going to study. Fine. Now I am going to list down the terminologies in the sequence which will be very easy for you to understand because it will be connected to one each other. Fine. Now, first genetical terminology is genetic itself. Fine. This is the first terminology. What is genetics? Genetics is a branch of biology which deals with the scientific study of heredity and variation. Point to be noted, I said scientific study of heredity and variation. So scientific study, why? Because even common person can say that the eyes of a child look like a father, the nose of a child look like a mother. Why not the nose of a child look like a father and the eyes of a child look like mother? Those reasons can be found out only by the scientific studies. That's why that scientific word is most important. The second terminology is heredity itself. What is heredity? Heredity is a transmission of character from one generation to next generation. The third terminology is variation. What is variation? It's a differences between the individuals of the same species, or it's a differences between parents and offspring of the same parents and individuals of the same species. In your textbook they have given it's a observable differences shown by the individual. Right? That is variation. Next terminology over here, that is a fourth terminology, that is gene. Gene you have already studied in the previous chapter, that is structure of chromosome. What is gene? Gene is a specific sequence of nucleotide present on chromosome coding for a particular protein which is expressed in the body. Fine? That is gene. Next terminology related to gene is factor. Factor was a term given by Mendel to the gene. Factor was term given by Mendel to the gene. Mendel called this gene as a factor. Fine? Now, what exactly is a gene? Meaning is particular segment of DNA. It's a particular segment of DNA. Now, friend, particular segment of DNA means what? All the DNA in your body does not called as gene. Only a particular segment which is transmitted from one generation to the next generation and express the character. That is called as gene. So factor is nothing but instead of particular segment of DNA, because during that time when Mendel was conducting the experiment, nobody was knowing about DNA, RNA, etc., etc. So he called it as a particle. Fine? He called it as a particle. Particle which is responsible for heredity and expression of character, that is factor. And this was a term by Mendel, don't forget. Next is, sixth number is allele. What is alleles? Alleles are nothing but alternative form of gene. It's a alternative form of gene. First half of the definition, which is present where? It is present on same loci of homologous chromosome. Now, what is homologous chromosome? It's a pair of chromosome. I hope you remember previous chapter I told you. Homologous chromosome, one from maternal side, one from paternal side. So, this is homologous chromosome. So, what is allele? Alleles are alternative form of gene present on homologous chromosome. Let's take an example. This is a gene, pink color, which I am drawing over here. It's a gene coding for eye color came from mother's side. On the next homologous chromosome, there will be the gene coding for eye color at the same loci. I mean that whether the chromosome is coming from father's side or mother's side, the gene will be at the exact similar location. Fine? This is called as alleles, and in Latin, alleles means friends. So, friends being together on the homologous chromosome, alleles. Very simple. Fine? Next, we have definition which is known as, terminology which is homozygous. Homozygous is also known as pure or pure breed. Fine. Now, what is the meaning of homozygous? Homozygous means when two similar alleles express character. For example, if height is tall, capital T is expressing character, then it is called as homozygous condition. And if it is small t for dwarf, short height, then also it is called as homozygous condition. The only thing you have to remember, student, when both the alphabets are capital and when both the alphabets are small, this is known as homozygous or pure line. Why pure line? Because pure line or pure breed means they will express only one type of character if they undergo self-pollination every now and then. Next we have heterozygous, or it is called as hybrid. So, heterozygous, another term is hybrid. Now, what is the meaning of this? If upper one is similar alleles expressing the character, then over here, dissimilar allele expressing the character known as heterozygous. Fine. Now, speaking about next terminology, 9th and 10th. They are opposite to each other. Over here, student, these three forms a pair, combination. These two forms a combination. 9th and 10th will form a combination. Over here, dominant and recessive. I hope you know what is the meaning of dominant and recessive. Dominant means which can suppress others, and recessive means which gets suppressed. Fine. So, you tell me, student, very simple. Can I explain dominancy and recessiveness in homozygous or heterozygous condition? Point to be noted. We cannot express or explain dominant or recessive in homozygous condition because both the alleles are same or similar. Fine. Hardly there is any difference. Next, in heterozygous condition only we can express dominant and recessive. So over here, with the help of this example only, I am trying to explain you. Over here you can see one is capital T and another is small t. Capital T is dominant over small t is recessive. So what is the meaning of dominance? The alleles which express its character in presence of alternate allele. So capital T is expressing its character in presence of alternate T, that is small t. So in case of capital T and small t, the person will be tall. Why? Because capital T is dominant, fine, and the small t cannot express itself, that is recessive. So very simple, the allele which cannot express itself in presence of alternate allele or which gets suppressed is known as recessive allele. Next definition is phenotype. Now what is phenotype? The physical appearance of an individual is known as phenotype. Opposite to it is genotype, fine. What is genotype? The genetical constituent of an individual is known as genotype. For example, for a height of a person or for a height of a person in the population, I can write down capital T, small t or capital T, small t. All these three sets will indicate what? Height. So height's genotype, the genetic constituent of a particular character, that is height, is three possibility. That is known as genotype of a height. For the individual also, if I make out the complete genetical constituent of my body, that will be also called as genotype. So what exactly is genotype? It's a genetical constituent for a particular character or for a particular person's body, that is genotype. Phenotype, physical appearance. Can you make out a person is a male or female by physically looking at them? Yes, definitely we can make it out a person is male or female by actually looking at them. That is known as physical appearance, phenotype. Fine. Next we have Punnett square. Fine. What exactly is Punnett square? I will teach you when we study the experiment. Over there, I will tell you what exactly is Punnett square. Then we have homologous chromosome. Fine. Now, what is homologous chromosome? This is done in the previous chapter also, but as it is mentioned over here, I am doing it once again. What is homologous chromosome? A pair of chromosome, one from father's side and one from mother's side. That is known as paternal side and maternal side respectively, known as homologous chromosome. Next we have karyotype. Fine. What is karyotype? Karyotype is nothing but chromosomal setup of any individual. For example, if I have to give my chromosomal setup, there will be 23 pairs of chromosome. Fine. At the end, there will be XX. Why? Because I am a female. Fine. If there is a male, there will be XY. That is known as karyotype. Don't forget, karyotype comes in exam. Students feel that out of syllabus question. No, it's there in your textbook. They have also given a picture of it. Fine. Next we have definition number 15 and 16. Fine. That is monohybrid and dihybrid. You have to pay more attention to this. Why? Because we have to study experiment of monohybrid and dihybrid. Fine. Now what is monohybrid? Monohybrid is inheritance of a single character. It's a study of inheritance of a single character. For example, if I'm studying inheritance of only height, then it is a monohybrid experimental study. If I'm studying the inheritance of two characters at a time, dihybrid. Right? Then trihybrid 3, but you don't have trihybrid in your syllabus. So this is a terminologies or basics of genetics which need to be thorough. Fine. Any 2 or 3 they ask you in your exam. So be thorough with this. Now we are going to study the experiments conducted by Mendel. More than 200 experiments were conducted by Mendel, but out of that you have to study only 2 experiments. Before we study the experiments conducted by Mendel, we need to know which experimental material he has used. You must be thorough about it, but once again, fine, revise it. The experimental material used by Mendel, it was none other than garden pea. Fine. And what is the scientific name for garden pea? You have to write down, student, garden pea, because there are many type of pea, cow pea, crow pea, etc. Wild pea. Garden pea scientific name is Pisum sativum. It's a binomial name, so you have to underline after writing it, or else you will not get marks for it. Why he selected experimental material as Pisum sativum? Why not any other experimental material? The very first thing, the reason was it was grown in the backyard of the church or monastery where he was living as a priest, and where he was practicing as a priest. But there should be certain scientific reason for selecting any experimental material for conducting the experiment. Randomly, we cannot conduct experiment on any material. So over here, there were many reasons, out of which you have to study 3 to 4 reasons. The first reason, fine, many seeds were produced out of single seed. If many seeds are produced, that will give varieties to study. Fine. So that is why he selected Pisum sativum. Second reason, easy to cultivate. It is also portable, easy to cultivate. Fine. In a very less fertile soil also it can grow. It requires very less nutrient to grow, not only grow but flourish. Fine. Easy to cultivate. Fine. Next reason about Pisum sativum was that it has a large flower. It has a large flower which used to carry out self-pollination. But as the flower was large, we can easily carry out artificial cross-pollination. So large flower was basically supporting the artificial cross-pollination. Fine. What it was supporting? Artificial cross-pollination. What do you mean by artificial cross-pollination? The pollination done by man, I mean myself, right? If I am picking up the pollen grain from one flower and dusting on the stigma of another flower to get the desirable character, that is known as artificial cross-pollination. Fourth and the most important reason for selecting Pisum sativum, the characters expressed by Pisum sativum was highly distinct. Fine? Highly distinct means if a plant is tall, its height will be 7 feet. If a plant is dwarf, it will show a height of 3.4 feet. There was no intermediate character between them. So that is why he selected. This is the 4 important reason why he selected Pisum sativum as an experimental material. There are many more, but from your syllabus point of view, only this much is there. Right? So remember, many seeds, varieties were shown, easy to cultivate, large flower which easily carry out self-pollination, and also comfortable for carrying out artificial cross-pollination. Characters were highly distinct, that's why. It is observable character. We don't need any microscope, any other instrument to see it. Fine? So this is the characteristics of Pisum sativum. Now, Pisum sativum express 8 characters in total. Human being express how many? More than 2000. But Pisum sativum express 8 characters in total. Out of that 8 characters, 7 characters were studied by Mendel. So you have to jot down this thing. Characters studied by Mendel for his experiment. Right? And in the characters, we have dominancy and recessiveness. So the character can be dominant or it can be recessive. Fine, character can be dominant or it can be recessive. Fine. Now I am listing down the character. Now a very simple tip I will be giving you. Number 1 character. Start from stem height. Stem height shows 2 characters. One is tall, another is dwarf. One is tall, another is dwarf by heart simultaneously. But the symbols which are used in the experimental sums or experimental process, that is capital T and small t. Fine? No D. Initials of dominant character is used. In case of dominancy, we keep it as capital. In case of recessive, we keep it as smaller. Fine? Second, seed color and third, seed shape. Seed color, seed shape. Fine? Simple. Seed color, yellow is dominant. Again, I am repeating, yellow is dominant. So, capital Y, and green is recessive. So, for green, it will go small y. Fine. Next, seed shape. Seed shape, round is dominant. So, we are using capital R, and wrinkled is recessive. Wrinkled is recessive. So, we are using small r. Fine. Next, fourth and fifth. Fine. Pod color and pod shape. Pod color and pod shape. So, what is a pod color? Green is dominant in case of pod color. Don't get mistaken or confused. Look over here. Yellow, green, green, yellow. Over here. Fine. So, green is dominant. Yellow is recessive. Small g. Fine. Pod shape. Inflated and constricted. Inflated, that is capital I, and constricted, that is small I. Now the next is 6th and 7th. Fine. I will also tell you which character was not studied by Mendel. 6th and 7th is flower, color and flower position. Flower color and flower position. Now speaking about flower color, in case of dominancy, it can be either violet, that is indicated by capital V, or it could be purple. Fine. That will be indicated by capital P. But the recessive character will be only white. So if you are considering violet, then you have to write down small v, and if you are considering purple, then you have to write down small p. Fine. Now speaking on flower position. Flower position, eggshell is dominant, that is indicated by capital A, and terminal is recessive. Terminal is recessive, that is indicated by small a. Fine. So these are the list of the character. The character which was not studied by Mendel was not studied. Fine. By Mendel was seed coat color. Fine. Not studied by Mendel was seed coat color. Let's not go in detail about it. Only this much you have to remember. Fine. Now, speaking of pod. What is pod? Pod is that structure in which peas are present. So, you must have seen like this, peas are present. So, this structure is known as pod. Fine. Now, speaking about flower position. What is eggshell and what is terminal? Fine. Eggshell. If the flower is on the side, fine, it is known as eggshell flower. And if it is at the tip, then it is known as terminal flower. So this is all about the characters studied by Mendel, or the list of character pea plant is expressing. So what about the tip which I told you? So the tip is very simple. Stem, seed, pod, flower. You have to remember like this. Stem, seed, pod, flower. SSPF. Fine. So stem height. If the stem is there, on that fruit will come, fruit will give a seed, and the seed will be present in pod, and later in the next plant it will give us flower when we grow it. Fine. This is how I remember. Very simple. Seed color, seed shape, pod color, pod shape, flower color, flower position. Fine. Last, he didn't study. You don't need to remember this thing. Fine. This is for your information. Yes. This year we completed the characteristics of pea plant.